everyone, welcome to the Twisted DNA. Today we're going to talk about flow cytometry. Well, flow cytometry is a powerful technology that can simultaneously measure and then analyze multiple physical characteristics of single cells. So some of these properties that can be measured by this process includes cell size, granularity or the complexity of your cell count, protein expression, health of our cells, as well as the cell cycle stage. So using flow cytometry allows us to measure thousands of cells in a matter of seconds, and we can also measure multiple parameters simultaneously. And let's say if you're working with some kind of rare population of cells, it also allows us to detect those rare population. Okay, so most of the flow cytometer has three main components. That includes fluidics, optics, and electronics. Here's a little schematic of a common flow cytometer that illustrates all three parts of this instrument. And now let's take a look at each of these components in much more detail. Okay, so the first we have is the fluidic system, which is responsible for transporting samples from our sample tube to the flow cell. So what is a flow cell? Well, flow cell is a place where the sample is introduced to a sheath fluid. Now, what is a sheath fluid again? It is just a solution that runs in our flow cytometer. Okay, so once your sample gets passed to the flow cell is designed in such a way that when the sample is introduced to the flow chamber, the flow of the sheath fluid moves the cells and restricts them to the center of the sample core. And this principle is called hydrodynamic focusing. So what happens with the hydrodynamic focusing is that all of our cells that gets lined up in a single file line, just as shown in this picture over here. And once these cells are lined up, they are ready to reach an interrogation point where they will be analyzed further. So interrogation point is the main analysis chamber of the flow cytometer, where these cells move past a laser beam, and just passing this laser beam will give us a lot of valuable information about our cells. And how's that? Is the first round of information comes from the light scattering. So what happens is that when an individual cell reaches the interrogation point and the light hits these cells, and the scattering of the light is omnidirectional. So there is a detector that is placed just in front of this laser light that detects the forward scatter signal. Whereas there's another detector that is placed 90 degrees of the forward scatter detector that detects the side scattering from our cell. So the light scattering activity of cells depends on the cell's optical density and its makeup. What that means is that this forward scatter signal that we have will tell us that about the size of our cells. So let's say if we have larger cell, you're going to have a large forward scatter signal. Whereas the side scatter tells us about the granularity of our cells, which is just a fancy way of saying complexity of our cells. Let's say our cells have more stuff in their cytoplasm, then they're considered more granular and they're going to have large side scatter signal. Okay, so with this information, what we can do is we can plot a forward scatter versus side scatter plot. So on our x-axis, we can have forward scatter signal, and on our y-axis, we can have side scatter signal. So now what this will allow us to do is that the large granular cells will end up in the top right quadrant of this plot, whereas the smaller cells or debris will end up on the bottom left over here. Okay, so the next we have is the optical system that includes lasers, mirrors, filters, and now let's discuss how each of them function in flow cytometry. So oftentimes when we're working with cells, they might be expressing some kind of fluorescence proteins that can be used for analysis, right? These fluorescence proteins might include GFP or MJE, or we might even tag our cells with some kind of fluorescent antibodies. So what happens is that when these cells pass through the interrogation point, in addition to picking the forward and side scatter signal, it also picks up the fluorescence signal with the help of different lenses and filters that's placed inside the flow cytometer. Okay? And based on the wavelengths of the light, the fluorescence signals gets rerouted to their specific detectors, like what we see in this picture over here, where each of these signals are being directed to their respective detectors, where this red signal is being directed to the red detector, yellow signals being directed to the yellow detector, and similarly the green signals being directed to the green detector. Okay? So lastly, we have an electric system whose main function is to convert the signal from the detector into the digital signal that can be read by a computer. So what happens is that this picture over here shows us that when the cell enters the laser beam, there is an increase in the voltage pulse, right? As you can see over here. And when this cell is at the center of the laser beam, we see that there is a peak at this signal. And as the cell pass the laser beam, the voltage signal goes back to the baseline, right? So these voltage pulses gets quantified by signal processor into some kind of numerical value, which are then transferred to the computer, and then this allows us to understand or read our data more efficiently. Okay, so that's it about the instrumentation. Now let's spend a few minutes on how to analyze the data. So over here, in this example, let's say we have a cell that is stained with a fluorescent antibody that has a red signal. Okay, so the first thing that we can do in our analysis is plot forward scatter versus side scatter. So again, like we talked about it earlier, where the large granular cell will end up in the top right quadrant, and whereas the small cell debris would end up at the bottom left corner, right? Now, in order to exclude this debris from our analysis and only to select the cells, what we have to do is called the gating. So gating is basically you're drawing this kind of like line, this red line over here. We are creating this gate so that we are only selecting cells and excluding debris for our analysis. Okay, so after we have gated for cells and excluded debris, the next thing that we want to do is something called doublet discrimination. So what that means is that we only want to focus on single cells for our analysis. And in order to do that, what we can do is change our y-axis to forward scatter height, and this allows us to gate for single cells. Okay, so why that is that single cell is going to have a smaller area as compared to the doublets, where doublets will have larger area. Now we're here gating for only single cells. And the population over here are the doublets that we are excluding. Okay, so now we have excluded debris and gated for single cells. The next thing that we can do is select only the live cells. So in order to do that, we can use several dyes, and in this example, we're using something called DAPI, which most commonly used to stain the dead cells. This way we can also gate for live cells and exclude dead cells from our analysis. After we've done this, what we can do is we can plot a histogram where the y-axis shows how many cells in our entire population have red fluorescence. Okay, and in the x-axis, we have the intensity level of that signal. So for the right, we go from the graph, the brighter the signal is, and vice versa. Okay, so here in this example, we have about 20 to 150 cells that's expressing red fluorescence protein. All right, so now let's look at another example. Again, let's say that we have cells that are stained with fluorescent antibody that has a red signal, but in addition to that, this time our cell also expresses blue fluorescent protein. So the initial analysis will still be the same as the previous example, where you exclude debris, select single cells and live cells. In addition to those analyses, here we can use something called scatter plot, as now we're working with two parameters, right? RFP and BFP. So for that, what we can do is we can have BFP on the x-axis and RFP on the y-axis. So Q1 and Q4 represents the population that's strongly expressing only one of the color. So Q1 represents the population that expresses red fluorescent protein, whereas Q4 represents the cell population that's expressing only BFP. Over here in Q2, these cells over here are called double positives because they're expressing both red fluorescent protein and blue fluorescent protein, so they're called double positives. Whereas the population in Q3 are called double negatives because they're not expressing either of the fluorescence, right? No red fluorescent, no blue fluorescent protein. So they're called double negatives. So this is also one way that you can analyze your data and get the idea of what percentage of your cells in your entire population is expressing certain kind of protein, right? So one thing that I would like to talk about is the fluorescence compensation. So what happens is sometimes when we're performing multicolor fluorescence studies, we have to be aware that there is a possible spectral overlap between fluorophores. So in this example, let's say if you're working with FITC and PE as our fluorophores, then what happens is that it predominantly signs off in the green emission ranges, but it also have photons that emits in longer wavelength that would actually end up in PE channel. Okay, so here we see that there is a spectral overlap and this would interfere with our analysis. And in order to mitigate that, what we can do is we can use something called compensation beads, where you only run single color control in the flow cytometer, and this allows the flow cytometer software to differentiate between these two signals and correct the background due to spillover. Okay, so that's it for this video. I hope you like it. If you do, please don't forget to comment, share, subscribe, and I'll see you all next time.